In this video, we will study about translation, which we will do together for prokaryotes and eukaryotes because there are not much differences between the two. I would recommend that you watch the transcription and replication video before you watch translation so that it is very clear and you follow step-by-step pattern. So first let's go into the basics and then we'll jump into the main process of translation. So central dogma of biology was given by Crick, and it is actually a kind of principle that how genetic information flows, which is always in one direction, that is DNA forms RNA by transcription and RNA then forms protein by translation. So this is the direction which is always one-way path. This was given by Crick. So already we have done how DNA forms RNA in the transcription videos, and now in this we will be studying how protein will be formed from RNA. Now let's first understand the kinds of RNA that are produced in transcription. So we already know there are three types of RNAs that are produced. One is messenger RNA. This RNA actually makes the protein. So messenger RNA is the one that makes protein, but tRNA and rRNA, they do not make protein. They assist in the protein formation. They have their roles in the formation of protein, but protein is mainly made by messenger RNA. It means whatever is the genetic information on messenger RNA, that way the protein will be formed. Protein is always formed, or translation occurs in the cytoplasm, whether it is prokaryotes or eukaryotes, it will occur in the cytoplasm. And for it, ribosome is an essential part. Now what is ribosome? These are cellular structures which synthesize protein. So ribosomes are also known as the protein factory of the cell because here is where protein is formed. Now let us understand the structure of ribosomes. Ribosomes have two subunits. One is the larger subunit and the other is smaller subunit. In ribosomes, these two units are different in prokaryotes and eukaryotes. So later we'll understand that, but first let's understand what is it made up of. So these ribosomes are made up of ribosomal RNA and proteins. Whether we are talking about the larger subunit or we are talking about the smaller subunit, both contain ribosomal RNA and proteins. And if we go to NCERT, it says structural RNA and 80 types of proteins are present in the ribosomes. Now these ribosomes are formed inside the nucleus and more specifically inside the nucleolus. So what happens is that after transcription, when ribosomal RNA is formed, then it collects these proteins which are inside the nucleus. When we are talking about eukaryotes, they collect all these proteins which are circulating inside the nucleolus, and then they form these large and small subunits. They are separate, they do not combine together in the ribosome. So these two subunits are formed separately and then they leave the nucleus through the nuclear pore into the cytoplasm, where they remain separate and only combine when they have to form the protein. So when they are separate, then these ribosomes are called inactive form of ribosomes. They are not forming any protein, and that is why they are separate. And when they join together, then they are called active ribosomes. So they join in the cytoplasm whenever protein synthesis is required. Otherwise in the cytoplasm also these subunits will float separately when protein synthesis is not required. So it is a kind of protective mechanism. So they remain separate, they are inactive, and whenever they have to form protein, then they join together and become active. Now in prokaryotes and eukaryotes, these ribosomes are different. In prokaryotes, ribosomes are of 70S type, and in eukaryotes they are of 80S type. Now what does S stand for? It stands for Swedberg unit. So now what is Swedberg unit? You have to just know the name, that S stands for Swedberg, but still. In prokaryotes, when these ribosomes are put in ultrafuge machine and when they undergo centrifugation, then they sediment. So the rate of sedimentation means how they are settling at the bottom. That rate is is calculated as Swedberg unit. So when we are talking about the larger subunit, it sediments at 50s rate and smaller at 30s, and together then they are at 70s. So that is why we cannot add and make it like 50 plus 30 is equal to 80. That's not the case. Here it is actually how they are settling at the bottom after centrifugation. So how they are settling together, then it is 70s, and if separate subunits we are talking about, then it is 50s or 30s. In eukaryotes, the sedimentation rate is different. That is why it is 80s. So that is 80s, S stands for Swedberg same, and the larger subunit at 60s, and smaller subunit will sediment at 40s. So these are the differences in the ribosomes of prokaryotes and eukaryotes. Now let us understand the basic structure and the basic information about tRNA molecule. tRNA molecule was first of all recognized by Francis Crick, and he called it an adapter molecule because he observed that this molecule is the one that is reading the code on messenger RNA and also binding to specific amino acids on the other hand. So this is what he observed, and that is why he called it an adapter molecule. He also called it sRNA, where S stands for soluble RNA. So when it was actually known that there is a molecule which is reading the code on messenger RNA and attaching amino acid, then it was called adapter molecule and also soluble RNA. Later, Robert Hawley gave the structure of tRNA. He observed and he explained the structure of tRNA molecule, and he said that it is a clover leaf shaped. So he gave the clover leaf model, which is a 2D model, and after that later another scientist gave the 3D model which was shaped as inverted L. So it is often asked that what is the shape of tRNA molecule. So if it is asked what is the 2D shape, then you'll say clover leaf model, and 3D is inverted L. Now when we look at tRNA molecule, we find that what is clover leaf, you can see that there are certain areas where this is rounded and here they are joined. It is because of the base complementarity. So wherever the bases are complement to each other, they form a straight arm like this, and wherever they cannot, then they turn and form this clover leaf type of structure. Then this end is known as 5 prime. So this end is 5 prime and this end is 3 prime end of tRNA molecule. 3 prime end is the end where amino acids are attached. So 3 prime end is the amino acid attachment site. When we look at the bottom, here it is written anticodon. So let's first understand codon and anticodon. Codon are specific sequences of 3 consecutive nucleotides, the triplets, on DNA or mRNA. So because it is a sequence of 3 nucleotides, which is called, that is why it is a triplet, then it is called codon, and it is on DNA and RNA. So if this triplet is on DNA or mRNA, then it is called codon. For example, if there are 3 nucleotides like AUG, adenine, uracil and guanine, this is a set of 3. So this is a codon. If it is on, we are reading on messenger RNA. If it is like CUU, so these are triplets and these are codons. Whereas anticodon is again sequence of 3 nucleotides, but it is on tRNA. So it is on tRNA, the sequence that is known as anticodon. So here is anticodon, which is from 5 prime to 3 prime direction, because you can see here that this end is 5 prime, this end is 3 prime. So when we read it GAA. So this is the anticodon. So every tRNA molecule has an anticodon, and on the basis of that anticodon there is a particular amino acid that they are carrying. And because there are 20 amino acids, there are for every amino acid there is a specific tRNA molecule. So there are many tRNA molecules. Then there is an initiator tRNA. Now by the word initiator tRNA, we can say somebody that initiates. So this tRNA is called initiator because it initiates translation, starts translation. Is there any tRNA to stop translation? No, there is no tRNA molecule to stop translation. We will study more on codon and anticodon and genetic code in a separate video. Now let's understand further. Now first let's look at the definition of translation. We have already discussed in central dogma that translation is formation of protein from messenger RNA. That is the basic definition. Then the next, if we want to give more precise definition, it is the process of conversion of nucleotide sequences in the messenger RNA into the sequence of amino acid to form protein. It means whichever nucleotide sequences are present on mRNA, according to that the sequences of amino acids will be formed to form protein, or process of polymerization of amino acids to form polypeptide. Polypeptide means protein, because protein has many peptide bonds, so it is also called polypeptide chain. So it's the process of polymerization of amino acids to form a polypeptide. So we can give these three definitions. First is formation of protein from messenger RNA. This is the simplest one. Second, process of conversion of nucleotide sequences in messenger RNA into a sequence of amino acids to form protein. And another is the process of polymerization of amino acids to form polypeptide. Let's study more about messenger RNA. I have already described tRNA and ribosomal RNA. Now let's understand mRNA. So like for transcription, there is a transcription unit. Similarly, for translation, there is translation unit which is present on messenger RNA. They have four areas. First is start codon. Start codon is the area where the translation starts, and the codon is AUG. So AUG is the codon that starts the translation, and the amino acid that is coded for it is methionine. So here is where the translation begins. Next is the region coding for polypeptide. So this is the region that codes for polypeptide. So whichever codons are present in this area, they will code for particular amino acid, and then the chains will be formed. That is, all these amino acids which will join with a peptide bond, and then the protein will be released, or polypeptide will be released till here. When they reach here, there is an area which is known as stop codon. Stop codons are three. They are either UAA, UAG, or UGA. So these are three types of stop codons. Any of the codons are present in this area, that is the point where the translation will stop and the polypeptide chain will be released. Next is UTRs. UTRs are untranslated regions. Untranslated regions. These are present before the start codon, that is this area. Here UTR is present, and after the stop codon. This is also UTR. So if it is asked where are UTRs present, they are present at both 5' and 3' end, but it is present before the start codon and after the stop codon. So this is about messenger RNA. Now let's understand the differences between prokaryotes and eukaryotes, how they are different in translation. Okay, now what are the differences in the translation between prokaryotes and eukaryotes? There are not much differences, only the difference is that the ribosome in prokaryotes is 70S, and in eukaryotes it is 80S. Then the other thing is that in prokaryotes, the messenger RNA does not have any cap or tail, whereas in eukaryotes there is a cap and there is a poly A tail. So these are the two differences. Okay, now let's understand a little more about ribosome before we start the main process of translation. The larger subunit of ribosome has three parts. One is A, P and E. These are the gates. There are 3 gates in this ribosome, A, P, E. A site is the one which is for the entry of new tRNA molecule. So here actually new tRNA molecule enters. P site is where the tRNA with growing polypeptide chain is present. So this site, a growing polypeptide chain is formed. And E site is the one where tRNA molecule after delivery of amino acid exits the ribosome. So here actually tRNA leaves. So there are 3 gates, we can say A, P, E. You can remember with ape. So with ape, A is the site where the tRNA enters, P is the site where the polypeptide chain grows, and E is the site where there is exit of the tRNA molecule after it delivers the amino acid. So I hope you have understood this. Now let's start with the main process of translation. Now let's dive into translation. Translation has 3 steps like transcription: initiation, elongation and termination. We know what is translation. Translation is formation of protein. And how is protein formed? When amino acids are joined together by peptide bonds. So these are the amino acids which are joined by peptide bonds, and then protein is formed. So initiation is same, starting of translation, starting of protein synthesis. Elongation is when this amino acid chain is elongated or the polypeptide chain is elongated, and termination is when it stops and polypeptide chain is released. So these are the 3 main steps. First step is initiation, which is further divided into activation of amino acid and transfer of amino acid to tRNA. So let's first understand what is activation of amino acid. We know that this whole process is going on in the cytoplasm. Translation occurs in the cytoplasm, and cytoplasm contains all these amino acids. We know that for protein synthesis we need amino acids, so they have to be activated. So for the activation of amino acid, 3 things are required: amino acid, there is an enzyme, and ATP. These 3 things are required. The name of the enzyme is amino-acyl tRNA synthetase. So this is the enzyme that actually helps in the activation of amino acid. We know that there are 20 amino acids, and so there are 20 amino-acyl tRNA synthetase enzymes. So these enzymes are specific, that is, for particular amino acid, the particular amino-acyl tRNA synthetase is allotted. So what happens is that this enzyme tries to find its amino acid for which its job is allotted. So here is the amino acid, it recognizes it, and then in the presence of ATP, it forms a complex. Now what is ATP? ATP is adenosine triphosphate. So it uses this ATP to form a complex. Now what is the name of this complex? As this is energy consuming, what happens is that 2 phosphate bonds are broken. So what is left? AMP, adenosine monophosphate. Only 1 phosphate group is attached, and that is why it is known as AMP after this complex is formed. So here is the amino acid and the AMP is attached, and there is this enzyme complex. This is formed. So if we write the whole equation, we can say this is amino acid plus ATP. In the presence of this enzyme, I am not writing here, but you know the name of the enzyme, it is amino-acyl tRNA synthetase. It forms amino acid AMP enzyme complex. This process of amino acid forming this complex is called activation of amino acid. Now this amino acid is activated. So this is the complex and it has activated the amino acid. Now let's go to the next step. Now after this amino amino acid AMP enzyme complex is formed, after that it will look for a specific tRNA molecule. I already mentioned that these enzymes are also of 20 types. They recognize a specific amino acid and they also recognize specific tRNA which has the specific anticodon. So, which will match with this amino acid that it is carrying. So, as soon as it finds its tRNA, then it binds to this tRNA molecule. So, now this has amino acid AMP enzyme complex. After it is attached, now the enzyme, its role is over. So, this enzyme is going to be released because it has to find another amino acid for the next protein or for the next tRNA. So, this will be released and even AMP will be released, and what is left? tRNA with this activated amino acid. This is known as charging of tRNA. It has got this activated amino acid and this is called charging of tRNA. Now, let us understand how this protein is formed. First of all, let us understand mRNA. This is mRNA. This is the start codon. Start codon is AUG. Okay. This is stop codon, which is UGA here. It can be UAA or UAG. Any stop codon can be there here. And the region between that is the region that codes for amino acid. So, this is the coding region. Okay. The area before the start codon is UTR, untranslated region. It means it will not code for any protein. And after stop codon, this is also UTR. This will also not code for any protein. It means only the protein will be coded according to the codons which are present in the coding region. Okay. So, this is mRNA. If we are talking about eukaryotes, it has a cap and it also has a tail. If it's prokaryotes, there is no cap and tail. Okay. The translation is similar in prokaryotes and eukaryotes, only the difference is of the cap or the tail which is present in eukaryotic messenger RNA. Okay. Now, let us first look at the definition of translation. Translation is the process of polymerization of amino acids to form polypeptide. It means there is polymerization of amino acids. Many amino acids are going to join together by peptide bonds, amino acids joined by peptide bonds, and protein will be formed. So, these amino acids will be joined according to the codons which are present on this messenger RNA. Whichever codons are present, that particular amino acid will arrive and peptide bond will be formed and this protein will be formed. This is the basic process that will happen. Okay. But let us understand how this amino acid is arriving and how this polypeptide chain is being formed. So, as soon as messenger RNA is formed and it is circulating in the cytoplasm, when it is ready and it is circulating in the cytoplasm, if we are just talking about eukaryotes, in prokaryotes they are already in the cytoplasm. But in eukaryotes, from the nucleus, when messenger RNA enters the cytoplasm, their ribosomes are already circulating. I had already explained the structure of ribosome, that it has two subunits, larger subunit and smaller subunit. Ribosomes are formed inside the nucleus, and when they are ready, when they are formed as separate subunits. They move from the nuclear pore and move into the cytoplasm and circulate there as separate subunits. That is about ribosomes. Now, they are already circulating as separate subunits, but as soon as this messenger RNA enters the cytoplasm, ribosomes come to know that okay, we are ready now, we have to do protein synthesis. So, the smaller subunit, this is the smaller one, the smaller subunit comes and attaches itself to the 5' end where the start codon is present. So this is the smaller subunit of ribosome. It comes and attaches itself where this start codon is present, AUG. So this is the first step. After that, as soon as the smaller subunit attaches itself, the tRNA molecule which has activated amino acid, means this is a charged tRNA molecule, it understands that it has to come and attach here. For tRNA molecule, we know that there is an anticodon loop. Anticodon loop has those bases which are complementary to this start codon if we are talking about starting off the polypeptide chain. So if this is AUG, the codon will be UAC on this. So this one is actually which tRNA? It is initiator tRNA. Initiator tRNA. So initiator tRNA is the tRNA that initiates or starts the polypeptide chain. So what happens is, as soon as the smaller subunit is attached on messenger RNA, this tRNA, this initiator tRNA comes and attaches itself to this area. So let me remove this. So it comes here and attaches itself to this area with its amino acid. So this is UAC. It is complementary, so it will fix itself. The amino acid that this particular initiator tRNA codes is methionine. So this is methionine. So it is, we can say AUG is the start codon and also it codes for methionine. So this tRNA comes and attaches here. As soon as this initiator tRNA comes and attaches here, the larger subunit of ribosome also comes and it attaches itself. Okay. How it attaches? We have already discussed that it has three areas, A site, P site and E site. So it attaches itself such that the P site contains this initiator tRNA molecule. So this much you have understood. Now let's move further. Always remember that initiator tRNA is the only tRNA which is present at the P site. Otherwise, all other tRNAs will always enter through the A site. Okay. Now, first of all, this initiator tRNA is at the P site. Now, the next codon which is on messenger RNA is UUU. So, the tRNA which is complementary to this codon will try to enter the A site carrying the amino acid. So, naturally, the anticodon will have the sequence AAA and the amino acid will be phenylalanine. It will enter A site. So, let's move next. We can see the ribosome has moved from 5' to 3' direction a little, so that this codon is in between the two subunits of ribosome. So, here there is already initiator tRNA on the P site and the next one has entered the A site. As soon as it enters, the amino acids between P and A site forms a peptide bond. These two sites are close to each other, that is why the peptide bond is formed. But who assists in the peptide bond formation? Ribosomal RNA, which is present in the ribosome, it acts as a catalyst and then it is called ribozyme. It helps in the formation of peptide bond. One is this and the other is that there is an ester bond present between the amino acid and tRNA molecule. So when this peptide bond is formed, that ester bond is broken and when it breaks, it releases energy which helps in the peptide bond formation. So these are the two factors which help in the peptide bond formation. One is the ribosomal RNA which acts as catalyst and second is how is the energy is given for peptide bond formation from the breaking of ester bond between tRNA and amino acid. After this, what happens is that this amino acid which is present at the P site moves towards the A site. So now we can say that when it moves towards the A site, this amino acid will go away and it will enter here. It means it will attach here on the A site. This tRNA molecule which is present at the P site is empty, so it will go towards E site. It has given the amino acid, it has no work to do, so it will go towards E site, and the tRNA molecule which is present at the A site, because now the P site is empty, it will go towards the P site. So this is the tRNA molecule containing 2 amino acids, and here is that empty one which will go away from the exit site. Okay. So this tRNA, first one, has left the ribosome because it has to look for another amino acid to form the protein chain for different AUG codon. So now we have this tRNA molecule which is present at the P site containing 2 amino acids. So you can see the ribosome has moved even further. We have moved towards the next codon. So tRNA molecule with anticodon GAA and amino acid which is leucine will enter A site. So now this is the amino acid, with this tRNA this has entered A site. As soon as this will enter A site, there will be a peptide bond formation between the 2 amino acids between A and P site. All these amino acids will go to the tRNA which is present on the A site. So again these 2 amino acids have left and they have attached to this tRNA molecule. The tRNA molecule which is present at the P site will move towards E site because now it has given the amino acid, and this will exit. Now this tRNA molecule which is present at the A site will go towards P site. So this tRNA molecule has now 3 amino acids. Okay, the last one has left here. So now ribosome has moved even further. Now it has gone to the next codon GUU. So the tRNA which is complementary to the next one will enter the A site. Here is the tRNA with this peptide bond formation. All these amino acids will go from P site to A site. And so here is the P site and A site. And so the last one tRNA, again this will be empty. All these 3 are going away. So now there are 4. 1, 2, 3, 4. So now this tRNA molecule will go from A site to P site. So this tRNA has now 4 amino acids attached. The last one will exit the E site, and then we go to the next codon. Same process, amino acid will join, and then finally again there will be 1, 2, 3, 4, 5. So we have taken an example of just 5 codons. So 5 amino acids, this polypeptide chain has been formed. Now this ribosome has moved farther end, and here there is UGA which is the stop codon. I've already mentioned that there is no tRNA for stop codon. So what happens is, as soon as this ribosome enters this stop codon region, a release factor, this is a protein, this arrives at A site. So this is the release factor, it arrives at A site. What does it do? It breaks the bond between tRNA and this amino acid, and this polypeptide chain is released. 1, 2, 3, 4, 5. As soon as this is released, both the ribosomal units will separate and this messenger RNA will dissociate. Always remember, the tRNA molecules, when they give the amino acid and leave the exit site, they again look for another amino acid. It means they can be used again. Ribosomes also, they separate and they again circulate. When they find a messenger RNA, new one to form polypeptide chain, then they will again be reused. But messenger RNA, once it has formed the protein, it will dissociate. Okay, so I hope you have understood how translation occurs, how ribosome moves from 5' to 3' direction, and the process is same in prokaryotes and eukaryotes. If you like my videos, then do subscribe to my channel and share and leave a comment. Thanks for watching.